This presentation is about non-invasive prenatal testing, or NIPT, as it is colloquially known. Technically, it should be NIPS because it's actually screening, not testing, but the term NIPT is still more frequently used in scientific literature than NIPS. So what are we talking about here? I'm going to go letter by letter here. So prenatal testing is basically all the different tests that are being undertaken for a pregnant woman to determine that the fetus is developing correctly. Invasive stands for diagnostic testing to confirm whether there are any potential problems with the fetus, and that requires collecting fetal cells, which means coming into close proximity of the fetus, which comes with certain level of danger to the baby. And N stands for non-invasive because NIPT test allows to potentially replace the invasive procedure to confirm if the baby is actually okay or not. So let's go a little bit into biology background in order to explain how non-invasive prenatal testing actually works. In a pregnant woman, there is an amazing structure called placenta that allows both baby and mother to coexist side by side without mixing their blood. And placenta keeps the baby's blood confined into a specific space, but baby's blood will be able to enter the placenta and then flow into these tree-like structures called chorionic villi, which themselves in turn are awash with maternal blood. So this allows maternal blood to come into very close proximity to the baby's blood without actually contacting one another. And the placenta itself is very thin. It's a thin layer of cells, thin enough so that molecular information between the mother's blood and a baby's blood can occur, so that mother can provide nutrition for the fetus, she can provide oxygen for the growing baby, as well as immunity, and in return, the fetus can exchange waste or carbon dioxide back into maternal blood. However, the cells that make up the placenta come from the same source as the fetus itself, basically the fertilized egg, which means the DNA inside those cells is the same as the fetus. And these cells, like any cells, some of them will die and burst, and they will release DNA into the local environment, which means the placental DNA, which is same as fetus DNA, will enter maternal blood, and this information can be collected.